Hello, and thanks for joining us today for the latest Technology Network webinar. Go with the flow, application of microfluidic 3D liver chip models for genotoxicity testing. I am Audrey Dubourg, Product Manager. For CN Bio, and I'm here to moderate the event. I am pleased to have Renato Cardoso and Talita Zanoni joining us today as your presenters. A warm welcome to you both. Dr. Renato Cardoso received his PhD in genetics from the School of Medicine at the University of Sao Paulo, Brazil. He then moved to the Ontario Cancer Institute for postdoctoral training. Dr. Cardoso transitioned to industry to lead the genetic toxicology department at Nucleotechnics. He is a diplomat of the American Board of Toxicology and a member of several toxicology societies. Currently, Dr. Cardoso is the Scientific Director of Genetic Toxicology at Charles River Laboratories in Montreal, Canada. He's leading the Genetic Toxicology Department and providing scientific and regulatory consultation for several industries. His research is focused on implementing human metabolic capacity in the test system used for genotoxicity assessment. He is migrating classical genetic toxicology endpoints to 3D co-culture models, as well as evaluating the use of NGS technologies to evaluate mutagenicity detection and mechanism of genotoxicity. Dr. Talita Zanoni received her PhD in genetic toxicology from the University of Sao Paulo, Brazil. As a researcher, she obtained international experience attending renowned academic institutions such as the University of Maastricht, University of Oxford, and the Vrije University Amsterdam, where she held a postdoctoral position. After a successful international academic career, which culminated in publications in high-impact factor journals within the realm of toxicology, she shifted towards the private sector by being appointed as a senior research scientist at Charles River Laboratories. Currently, her work focuses on the development and application of new 3D advanced in vitro cell-based assays as an alternative to animal testing in drug discovery. Following the webinar, we will have a Q&A session and we welcome any questions that you may have. You can submit questions for the Q&A session at any time during the presentation. To ask a question, you should enter your question into the box on the right-hand side of your screen and click Submit. We will answer as many as possible during the time available. For any questions we do not have time to get to, we will be sure you're contacted offline with an answer. Please remember, you can ask questions at any time, even whilst watching the webinar on demand. So, without further ado, I will now hand over to Renato.
Hi, Audrey. Thanks a lot for introduction. As Audrey just mentioned, my name is Renato Cardoso. I'm going to be presenting about our experience with the 3D liver chip models and how we're using it to generate new genotoxicology assays. I will start with a brief overview of what we understand that are some of the problems of genotoxicology battery. And then on the second half of presentation, I'm going to pass to Talita, and she will present on the results that we obtained in our laboratory. I will just start with a brief review of the concepts in genotoxicology, because I understand that some of you might not be genotoxicologists or might be new to the field. So in genotoxicology, we are concerned about the potential of agents, chemical or physical agents, to lead to the formation of DNA lesions. It's important for us to remember that DNA lesions might be different, may be arising from different mechanisms, and they may lead to mutagenicity, that is, changes in the sequence of DNA, clastogenesis, that are structured changes of chromosomes, so breaks or rearrangements, and those are the two most important for the genotoxicology assessment, because if a compound leads to mutagenesis or clastogenesis directly, then there's no recognition of a safe exposure level to that compound. There's also aneogenesis, that are changes in the number of chromosomes, and the epigenetic changes that might change the regulation of the genes. But those are not typical the base of the study for genotoxicology. So once there's a DNA lesion, there are different potential outcomes. The cell might just use its DNA repair system, repair the lesion, and continue. The cell may enter a state of senescence, where it stops proliferating and decreases its metabolic activity, or if there's too much of damage, the cell may die by apoptosis. What we're concerned the most is when none of those happen, and the cells actually perpetuate a change in DNA and may transmit that to the other cells. Because if that happens in gonadal cells, you may lead to the genetic syndromes, and in the case of somatic cells, we may have the occurrence of cancer. Also, genotoxicants might directly bind the DNA. Your compound might physically bind the DNA and change its structure, or the compound itself might be inactive in that sense, but when metabolized, generate DNA reactive metabolites that can be pre-genotoxicants. How different industries look to that, right, to the genotoxicology assessment? We have a requirement to have a multiple testing. So we work with a battery system because there's not a single assay that can detect all the different mechanisms of genotoxicity. So there's a slightly different requirement for the number of tests and which tests you can use, depending on the industry you work in. If it's pharmaceuticals, you typically have 3 assays. If it's medical device, it depends on how intrusive the medical device is. For cosmetics, for example, you cannot have tests done in animals for a lot of different countries. So it's important to consider what is the requirements. But as a common point, they all use more than one assay. And what that does for the assessment? If you think that every single assay will have a sensitivity and specificity, that means that when you work with a battery system, you're going to have an additive effect. So the more assays you do, the higher your sensitivity. So the combined sensitivity will allow you to detect more of the true mutagens and genotoxicants. But that comes at the expense of decreasing the specificity. And that actually increases the rate of misleading false positives. So this is not by accident, that's by design. And what that does is that it helps regulators to protect populations and the environment to be exposed to genotoxicants. But it comes at the expense of a burden to industry, because now the industry will have different compounds that are safer, and they will be erroneously identified as genotoxicants. So that particular company may decide to drop a compound that had a good potential. That we understand the concept of the battery system. One of the problems that when we look to these batteries, and this is example of a battery that would be commonly used for pharmaceuticals. We would use an Ames assay, in vitro micronucleus assay in mammalian cells, quite often in human lymphocytes, and then in vivo rat assay, for example, the in vivo micronucleus. In the in vitro assays, we use rat S9, and this is a liver fraction that you add to your culture system. The problem is that even if we work with human lymphocytes, the metabolism is all based on rodents. And the difference in the metabolic profiles may actually lead to different recognition of the promutagens. So you may now detect something that should be detected as a positive, as a negative, and vice versa. So if we think about the rat S9, there's pros and cons. Of course, there's pros, that's why we're still using it. So they're readily available, there's low lot-to-lot variability, they're well characterized, and quite often people will mention that they're induced, and that's a pro. But one thing that we have to question, right? Normally, when we induce something, when we induce certain categories of CYP enzymes, that comes to expense of decreasing others. So unless you know exactly which ones are important for the metabolism of your compound, this is not exactly good feature. And as a con, I think that the most important is that using a metabolic system that is not intrinsic to the cells, you're going to lose one of the most important advantages, that is the CYP enzymes can be activated by the compound that they metabolize, meaning now the cells are not going to increase the amount of CYP requested to metabolize the chemical that is being tested, because you're using a system that is not intrinsic. As an alternative, we can look to other cell types, right, that it has metabolic capacity, and see how we could use those for genotoxicology testing. So one of the most common alternatives to the rat S9 would be the use of HepG2 cells. There are some features, though, that are not very attractive on those. Those are hepatocellular carcinoma cells, so they are quite transformed, and it's well described that they have certain enzymes at very low levels or absence of the enzymes, especially in phase 2 metabolism. Then more recently, people have been using HepRG cells and reporting some very promising results with that. Those are not as transformed as the HepG2. They have more enzymes for the phase 2 and active receptors, transporters, so they're more desirable than the HepG2. One of the problems, though, is that they still have certain classes of enzymes like CYP1A2, 2A6, and others that are relatively reduced compared to the primary hepatocytes. So if we look now to the primary human hepatocytes, we're going to see that those are the golden standard. So they're commonly used for metabolism studies. And this is very good, because if you could use those cells in genotoxicology assays, we would have a close metabolic profile of the compound being analyzed. Now, they are also p53 competent, what is very good for genotoxicity assessment, and they will have active receptors, transporters. One of the problems, though, is that they lose their metabolic capacity very shortly after being put in culture. So you can't keep them as long because they're not immortalized cell lines. But if we look to other factors that influence the metabolic capacity in vitro, you're going to see that there are ways to extend this metabolic capacity further. If we migrate from 2D culture to 3D culture, if we add other cell types to the culture system, like the sinusoidal cells, or even if you compare a static culture with a culture under flow, since that the microfluidics actually help to promote and sustain the metabolic capacity of cells longer. So having that in consideration, we hypothesize that we can potentially increase the accuracy of the genotoxicology assays if we incorporate those features of human metabolism in vitro, and if we combine different culture systems and features like 3D, the right cell type, the co-culture with other cell types in this culture system. So migrating the classical genotoxicology endpoints from a common 2D screen to those more complex cultures might help us to increase the accuracy and decrease the rate of false positives. So now I'm going to pass the presentation to Thalita Zanoni, and she will cover some of the results that we're obtaining in our laboratories. Thank you.
Well, thank you, Renato, for your presentation. Now this table, I will give a brief comparison of the different complexity levels of the different types of existing cultures. So for instance, we have 2D cell cultures that when cells, it's the classic, when cells grow into a plastic, they are forced to adapt into this plastic. So as a result, for the hepatocytes, they can lose some of their metabolic capacity, which this does not occur. This occurs less when we're testing spheroids. So an advantage of spheroids when in comparison to the monolayer is that we can culture actually multiple cell types, such as hepatocytes or hepatocytes in endothelium. We also have the liver sandwich culture that allows us to culture a higher number of cells. So if you look at this table, we can culture about approximately 775,000, when the spheroids, they're made with approximately 1,000. And it also allows us to culture different types, multiple types of cells, such as endothelial. So we also have the open liver chips that they offer the same advantages as the liver sandwich, but the main advantage is that they have flow into it, while the liver sandwich does not. We also have the closed liver chips that offers the same advantage as the open liver chips, although they can be made with less cells, so approximately 235,000 cells, while the open liver chips, we have 775. And they are about approximately 7 times more expensive. But they always also offer the microfluidic flow. So in this work, we tried to adapt the use of spheroids, liver sandwich, and open liver chip into our genetic toxicology testing. So before I go ahead and show you the results, I will talk a little bit, very brief, about one of these tests that we try to incorporate in these 3D models. So for instance, in the genetic toxicology test battery, we have the micronucleus assay. So during the cell division, acentric chromosomes fragments caused by clastogenicity or whole chromosomes caused by anogenicity, they may not migrate correctly, forming a micronucleus separate from the nucleus of the daughter cells, as we can see here in the white arrow. So these cells, they are stained and evaluated for the presence of micronuclei. This assay requires cell division to detect DNA damage. We also have the COMET assay, which requires the single cell suspensions in agarose gel that are spread over microscopy slides. And after deproteinization, electrophoresis is run. Although the intact DNA molecules, they are too big to migrate once electrophoresis is run. So they remain on its original position of round nucleus, as we can see on the first photos here. While the small fragments of DNA, they migrate and form a smear. And this assay is called the COMET assay because, as you can see, the image resembles a lot like comets. So the rate of DNA contact in the nucleus and smear is calculated, and this assay does not require cell division to detect DNA breaks. So the micronucleus and the COMET assays, they may be used in conjunction with the Ames test to evaluate genotoxicity for several industries. So now I'm going to show the results we obtained here in our department with spheroids. So we created the spheroids using HEPA-RG cell lines. So we used 96 ultra well low attachment plates, and we kept them in culture for 10 days. Here is the photos showing the evolution of how they became, how we obtained the final spheroid on day 10. So after we obtained the spheroid, we decided to treat it for 24 hours with direct mutagens such as EMS and MMS, and cyclophosphamide, which is indirect mutagens. So we tested actually direct mutagens and indirect mutagens. After that, we gathered 10 spheroids, and then we had to create a single cell suspension that we used for the evaluation of DNA strand breaks using the COMET assay. So this table indicates that we obtained very acceptable values for negative control, such as high percentage of mean cell viability, low hedgehogs, and a very low percentage of tail intensity. While we treated these spheroids with EMS and MMS that are direct mutagens, we obtained a positive response. Although when we tested the indirect mutagen, cyclophosphamide, we did not obtain a positive response. So our conclusion for this assay, for the application of this assay to genetic toxicology testing, is that it sustains cell viability for up to 10 days, which is quite good, although it offers limited cellularity. That is, each spheroid was made with only 1,000 cells. So for metabolism studies, we will need way too many spheroids. There is a recent publication in the literature showing that this can be done, although for our purpose of testing, we found it very time consuming, and this would definitely increase the cost in the industry. And we found the test difficult to manipulate, or it would require the use of robots. So although spheroids are used to screen other endpoints that do not require a lot of cells, we did not find it efficient for our purpose. So thinking about this, we decided to move on to the 3D liver sandwich model. So we decided to make this test based on the in vivo morphology of the human liver. As you can see here, we have the hepatocytes and we have the endothelial cells. So using a 24 or 96 well plate, we decided to create a prototype that would better mimic the in vivo situation. So we put on the 24 or the 96 well plates. We covered it with extracellular matrix. Then we added the endothelial cells. After 24 hours, we seeded the hepatocytes. And after 24 hours, when the hepatocytes were nicely adapted to the plate, we added matrogen on top. This made the cells go back to their environment. And this in vitro prototype was later adapted to a liver chip system. So this is how we created this 3D liver chip sandwich models. So for instance, in a 96 or 24 well plate, we created the liver using HepRG cells. These were kept at 9 days in culture. And after that, we added TK6 cells that are proliferative cells on top. And then we obtained a ready co-culture system. This co-culture system was dosed just like the in vivo studies used in genetic toxicology. For instance, we dosed at 0, 24, and 48 hours. After this, the co-culture system had to be detached so we could actually perform on the upper part of the transwells that we have the TK6 cells, the micronucleus assays. And just remembering that these cells are proliferative cells, and for this, it allows the micronucleus assay detection. So the bottom part of the 3D liver sandwich model, we obtain the single cell suspensions and we run the COMET assay that evaluates DNA strand break. The hepatocytes, they do not proliferate, and that is why they are good candidates for the COMET assay. So to make this possible, we had to make sure that the cells were growing healthy with the cell culture medium we were using. So for instance, the 3D liver sandwich models, we use an in-house hepatocyte media, but we had to culture them with TK6 on top. And since TK6 are proliferative cells, we had to guarantee that this was still going on. So the adequate media for the TK6 cells to be cultured is the RPMI media. So you can see here that this keeps the TK6 cells with a high viability and the population doubling time at 12.5 hours. So when we cultured the TK6 cells on top of our 3D liver sandwich model, we saw that the cells died. So we had to look for other candidates of media, and then we decided to use the HepRG pre-induction media from Lonza. After characterizing the media, we measured some liver-specific physiological functions, such as albumin and urea, and we saw that there was an increase on them through time. Another important question we had is if there was communication between the two different compartments. So for instance, when we dose the bottom compartment, the liver itself, with MMS, that is a direct mutagen, once we added the transwells on top, if these damages were able to be induced within the TK6 cells. So our results show that yes, we observed an increase on those response after treating with MMS in TK6 cells, and there was also a dose responding increase regarding apoptosis. So here are our final results for the 3D liver sandwich. So here we can see that our untreated cultures for the COMET assay, they had a high viability, a low percentage of hedgehogs, and a low percentage of median tail intensity, while a very low percentage of micronucleus was also seen. Once we dosed MMS, that is a direct mutagen, we saw an increase on the percentage median tail intensity with the COMET assay, and also an increase on the percentage of micronuclear formation. Once we tested the indirect mutagen, BAP, we only observed an increase on the percentage of micronuclei, whereas when we tested cyclophosphamide, this was not observed, although there was a small increase on the percentage of micronuclei, it was not statistically significant. So our preliminary conclusion for the 3D liver sandwich culture is that it is one assay that allows the determination of two genotoxic endpoints, the COMET and the micronuclease assay. We obtain an ideal amount of cells that allowed us to perform both of these tests. And this test was able to detect genotoxicity caused by direct and indirect mutagens. We suggest, therefore, that the metabolism is active, but this system can further be improved. Thinking about that, we decided to test the open liver chips that offer a higher level of complexity because it has the flow. So we applied, using the same prototype, we applied microfluidics technology in the liver chips. But now we created the liver chips. And the difference is that we inserted flow into this. So the presence of flow, it increases media circulation, it also stabilizes the nutrient supply, and it also improves the waste metabolites removal, and it better controls the oxygen tension within the culture. So this is how we created our in-house liver chip. So we built the same prototype we used for the liver sandwich into these inserts, as you can see in the photo. The inserts were added to a microfluidic plate, 24-well plate, and they were incubated under flow at 37 for approximately 7 to 8 days. You can see on the bottom of the plate, there's microfluidic channels that allow that the cell culture media will flow from one well to another. And one of the main advantages is that the controller is placed outside and it can add flow pressure into the plates, mimicking human liver dynamics. So here we tested two different 3D liver chips, one built with primary hepatocytes and the other one with HepaRG. So I have here some images of the negative controls showing already that the flow really improved the cell morphology. Here we have the photos of the 3D liver chips, how the hepatocytes look on it. You can see that they have a nice hexagonal shape, typically characteristics of the hepatocytes. Once we also saw improvements on the cell morphology of HepaRG. Here, for instance, we have a photo of the endothelial cells before hepatocyte seeding. I'm going to show you now the results of the HepaRG using the flow. So we can see that the untreated cultures had a high viability, low percentage of hedgehogs, and also a low percentage median tail intensity. These values were also acceptable for the micronucleus test. Once we dosed the cell culture with MMS, we saw a statistically significant increase of the percentage median tail intensity in the COMET assay, and also an increase on the percentage of micronuclei. While we tested BAP, that is an indirect mutagen, we saw an increase in both of the tested systems. And in contrary of what we observed within the test of spheroids, we saw here, by testing cyclophosphamide, that there was an increase on the percentage of micronuclei. These are the results that we obtained with primary hepatocytes. As previously mentioned by Renato, the primary hepatocytes, they are the golden standard of metabolization for in vitro testing. So again, we had acceptable values for the untreated. We did obtain increase in the percentage of median tail intensity and on the percentage of micronucleus that were higher than what we observed with the sandwich liver cultures. Indirect mutagen with BAP, we also got a positive response. And finally, I can show you that we had higher values than when we used the HepaRG 3D liver chips, while the cultures were treated with CP. These results, they are in line with the in vivo study found in the literature, as shown by Recio, where he showed that in vivo studies showed that cyclophosphamide, it had no effect on the liver, but they obtained positive results in blood micronucleus. So this would be possibly an explanation. So our preliminary conclusion for the liver chips is that we are able to evaluate two genotoxic endpoints within one system. We also believe that the liver chips with flow will better recapitulate in vivo human metabolism, contributing to the decreased rate of misleading positives in genotoxicity assessments. The system is in line with the 3 Rs, that is reduce, refine, and replace. And we also believe that they could potentially be a good alternative to animal testing. So thank you, everyone. We are open to questions.
Thank you very much, Renato and Talita. I'm sure you'll all agree that there were some excellent points raised during the presentation. We will now begin the Q&A session. So to submit a question to our speakers, you should enter your question into the box on the left side of your screen and click send. We will answer as many questions as possible during the time available. We have had several questions submitted already, so I'll now move straight on to these. So the first question is, what first attracted you to the CN Bio system?
We do acknowledge that nowadays it's quite difficult because there are so many systems that require this flow into the market. But one of the things that, if I think that we really was that it seemed an easy plate to use, and because it was an open plate, this was very it seemed easy. That's what first attracted us.
Okay, excellent. Thanks, Talita. So the second question is, how do you view CN biotechnology compared with other organ-on-chip technologies?
Well, again, what I just said before, there are many things. And we did actually have experience with different plates and with different systems. But what's really attracted us with the CN Bio system is that because it's an open plate, it's relatively easy to use. And to our purpose, that we required a certain amount of cells, it was very effective. And also another thing that attracted us was the cost, because the cultures, they would actually, the price of the cultures would actually make more sense to create a method that could be commercially available. Let's put it that way.
Okay, excellent. Thank you. The next question we have is, since there is a current demand on decreasing the use of animals in science, I'd like to know if your department offers other 3D model assays that could serve as an alternative to animal testing.
That's actually an interesting question. That is one of our research interests here within our department. So as you've seen, of course, the presentation we just gave, but we also have other things that we're trying to evaluate. So we recently moved on with working with the 3D skin models. And we actually now are able to offer already GLP comets using 3D reconstructed full thickness models.
Okay, excellent. Thank you. The next question is, how long was the exposure period for these samples?
We actually use the dosing periods exactly like the in vivo studies for genetic tox. So we dose at 0, 10, 24, and 45. And it's important to dose at 45 and collect the cells at 48, because then when there's the repair time, the cells do not have time to repair. So then we could actually measure the damage.
Okay, thank you. The next question is, what are the setup differences between liver sandwich, open liver chip, and closed liver chip?
Actually, the liver sandwich was a prototype that we created because we wanted to see if the metabolism would be better because of the interaction with different cells. So we actually started investigating there, but there is a chance, because there's not flow into it, there is a chance that the hepatocytes, they still would lose their metabolism, their CYP enzymes active, the activated CYP enzymes. So the closed one and the open one, they actually function kind of the same. They both have the flow, but the only difference, what we found is because one, we could culture more cells than the other. So for our purposes, I know that both are very good, but for our purposes, we prefer to use the one we used.
Okay, thank you. So moving on, the next question is, apart from albumin and urea, what other parameters have you investigated?
Sorry, I think I got a bit disturbed. Can you please repeat?
That's okay. So apart from albumin and urea, what other parameters have you investigated?
We have initially started investigation other CYP enzymes like CYP1A1, but we're still moving on to more testing. In our further testing now, what we're really interested in doing is actually amplifying the amount of pro-mutagens of test compound positive controls, so that way we can actually see how effective the system is. We're pretty confident, but we will see further. We will have further results.
Okay, brilliant.
May I actually chime in on this question?
Sure.
We also we actually tested the expression of the CYP enzymes itself. We reproduced in the experiment, so we found that the results were quite preliminary yet to be shown, but we are quite happy with what we're seeing so far. But the idea was that we wanted to actually test several CYP enzymes and have sort of a metabolic profile of the induction of the CYPs. The other thing that we had planned is the bilicanalicular staining, just to show morphology of the cultures, but we are looking specifically to applications in genotoxicology. So I think that for us at this point, the most important is understanding if this system is being able to metabolize the promutagens, and if those promutagens would be detected as mutagens as they should in the genotoxicology battery. So we are focused more on the indirect evidence of metabolism as the conversion of the promutagens.
Okay, thanks Renato and Talita. The next question we have is, do you typically compare and validate in vitro results with mouse assays or human data, clinical data?
That is a very good question. We don't typically look, well, how can I put that? That is still on the first days, I would say. We still have to do a lot more work to be able to talk about clinical data, so we are not looking to the human data right now. But what we are doing is compare for now with the results that we have in vivo. So a lot of things, for example, when we have the positive in vivo, it's very hard to move things on development and get clinical data. So we could look to the clinical as more of evidence for the negatives and help us to conclude that if something is really being detected as a false positive, but you would not typically see a lot of positives in the human data. So for now, we're focused more with the comparison with the mouse assays, and I think that the case of this cyclophosphamide evidences well that this we've been able to detect some particularities of the in vivo testing with this liver model.
Okay, excellent, thank you. The next question is, have you tested the difference between the HepA-RGs and primary hepatocytes in the other models, for example, the spheroids?
We actually did not. The spheroids, we started with the HepA-RG, but since the principle of the test would be the same, we thought it wouldn't be, we straight away took the decision that we would stop there and then we would move on to a different test model.
Yeah, just to add something, we really believe, and we saw several, we actually have been in communication with several other teams that use the steroids for other purposes. Steroids are great, they work well, but it depends on what's the application that you're testing, right? And for a GLP study, we just found that it would be a little bit too convoluted to invest on the steroids, as we would need to pool the steroids to have enough cellularity to test things like a micronucleus or comet. And the decision to move away from steroids was not because they don't work, but just because we felt that for the application, it would be more practical to move with an open chip.
Thank you very much, Renato and Talita. That's all we have time for today. And just to remind everybody that any questions we didn't have time to get to will be answered offline as soon as possible. And you can continue to ask questions even when watching this webinar on demand. The webinar will now be made available on demand. If you have any friends or colleagues that you think may be interested in this webinar, please feel free to share the link. And thanks again for your time.